With this video, we're going to be looking at the eyes. This is going to be exercise 23 and 24 in your lab book. So go ahead and get that out and turn to page 352. Feel free to stall the video while you get to the right page. So figure 23.1, don't look at it. You can ignore that picture. I'm not going to show you a model that resembles that whatsoever. Figure 23.2 shows you the muscles of the eye, which we will see, and 23.3 shows you the eye. So again, we will be looking at that. So let's go ahead and get started. So this is the eye. This is going to obviously be just looking straight at you. The top muscle here, that is going to be the superior rectus. This bottom muscle is going to be the inferior rectus. Now, when we look at the eye, the easiest way to tell where the medial or lateral side is to look at the top, at the superior oblique. The superior oblique tendon points towards the nose. You'll see it in a different shot. It's right here. So that means the nose is over here. So that makes this muscle the lateral rectus, and this muscle, this muscle the medial rectus, and this muscle the lateral rectus. Now we have the sclera. The cornea would be right here. It'd be the clear part in front of the iris. This is the iris, and that is the pupil or the opening. So here we've taken a side shot of the eye so we can see it a little better. So we have the superior rectus muscle, the inferior rectus muscle. This is the inferior oblique and the superior oblique muscle coming this way. So that makes this the medial rectus. And again, the sclera, the iris, and the pupil. This is from the other side. So again, superior rectus. We have the inferior rectus. We're going to have the inferior oblique. And you can't see the superior oblique from this angle. And this is going to be our lateral rectus muscle right there. Again, we have the sclera. The clear part would be the cornea. We have the iris, and we have the pupil. Taking off the top portion, this is the choroid. The choroid is the dark layer of the eye. Its job is to absorb excess light coming in so it doesn't bounce around on the interior of the eye, causing blurriness. We can still see the iris and the pupil, and the white part again is the sclera. So here we've taken apart the model completely. So we have the sclera, the superior rectus, the superior oblique, the lateral rectus, and the medial rectus. We have the choroid, and we can see the optic nerve right here. And we have the iris and the pupil. This is the lens. The lens would sit right here in this little dent in the model. And again, we can still see the sclera, the iris, the pupil. The choroid is over here. This area is the retina. This is where we have our photoreceptive cells. This is the optic disc, or the blind spot, where we cannot see because this is where all the axons are leaving the eye. So there are no photoreceptors here. That is our optic nerve. And this is going to be our macula luta. And in the very center, we're going to have the fovea centralis. Again, we've moved on, and we're at figure 23.3 in case you're a little lost. When we look at figure 23.3, you can ignore picture B, the one that looks like neon rings. We're not going to look at that view. So picture A is what we are concerning ourselves with. Now, on that picture, you do not have to know the anterior pole. So that's going to be by the spiral, the sixth word down, anterior pole. And we do not have to know the posterior pole, which is on the free side of the paper. The second from the, it's right above the optic nerve word. So cross out those two things. Other structures that we can see here, when we look at our model compared to our illustration, we're going to have the ciliary body. That's this part right here. It kind of looks like a mushroom cap if you look at it from the other side. When we look at the spaces in the eye, everything in front of the lens is going to be the anterior cavity. Your book says segment, but go ahead and cross out the word segment and write in cavity. And back here is the posterior cavity. Again, your book says segment, cross out segment, write in cavity. The posterior cavity is going to be filled with your vitreous humor, your eye jelly, and the anterior cavity is going to be filled with aqueous humor. It's more watery. Now, the anterior cavity again is divided into two areas. We have the anterior chamber in front of the iris and the posterior chamber behind the iris. Super close up again, so we can see kind of more of the interior of the eye. We have the sclera, we have the choroid, this is the retina. We have the optic nerve and the optic disc or blind spot. We have that macula lutea. In Latin, macula means spot, lutea means yellow. In real life, it's kind of a yellowish color. This area has no rods, so it's only cones. And in the very center of that macula lutea, we have the fovea centralis. That is where we have the highest number of cones, and that's where our best vision is located. This is that ciliary body, again, looking kind of like a mushroom cap. This is where the lens would have been sitting. Okay, so it's right there now, but it would have sat there. So again, from the lens to the back, that's going to be the posterior cavity. The lens forward is the anterior cavity, and the anterior cavity is divided into two. The anterior chamber in front of the iris, so cornea to the iris, and the posterior chamber from the iris to the lens. Okay, just a slightly different view of the same picture. So stuff that would have normally been written on the board for this chapter, we need to know the bones of the orbits. So this would just be strictly a word question. So I might ask list the bones of the orbit or which is not a bone of the orbit, that kind of scenario. So we have the frontal, the zygomatics, the maxilla, the lacrimal, the sphenoid, and the ethmoid. The caruncle. The caruncle is going to be a combination of sebaceous and sweat glands. It secretes that whitish material that collects in the corner of your eye, especially if the eyes are dry. The secretion is going to, one, prevent the eyelid from sticking to the eye itself. That sebaceous gland, that oil gland, is going to create an oil slick so nothing sticks together. We've all know that if you wet something, sometimes they stick, kind of like a wet t-shirt sticks to your tummy. So if you did not have the caruncle secretions, your eye would stick to the eyelid. And the sweat glands and the sebaceous glands are going to also help wash away debris and kind of create an antibacterial environment. This is why if you cry a whole lot, sometimes your eyes get red and irritated and dry feeling. Your tears wash away the secretions of the caruncle and your eyelids start sticking to your eyeball. That's with the dry feeling and the irritation feeling that you get when you wash away this material. The plica semilunaris, so again the term plica means fold and semilunaris means half moon shaped. This is that pink flap at the corner of your eye, a medial corner of your eye. It is homologous to the nictitating membrane of reptiles and frogs. So homologous means that originally was the same but now we're different. So the nictitating membrane of a frog or an alligator, that's the clear membrane that covers the eye when they're underwater, so water doesn't actually touch the eye itself. Ours obviously doesn't do that anymore. It's something that we, since we don't swim underwater, we're not aquatic, we don't need anymore. So all we have is this little fleshy piece of skin there. In cats, this is that part that whenever they start to close their eye, sometimes they've got that medial fold that kind of starts creeping across their eye. That's the same structure. Theirs is more mobile than ours is. Okay. So here's where we were talking about the cavity. So again, the anterior cavity is the same as the anterior segment. Your book likes using segment. I prefer the term cavity. I'll never type the word segment. So again, the anterior cavity is from the lens to the cornea. The posterior cavity is the same as the posterior segment. That is from the lens to the retina. And that's going to have vitreous humor where the anterior cavity has aqueous humor. And as I mentioned, the anterior cavity is divided into two. The anterior chamber is from the cornea to the iris, and the posterior chamber is from the iris to the lens. So you can kind of think of the chambers kind of like rooms in a house. So chambers within a cavity. The macula lutea, again, on the model it appears as a pink spot, but in real life it's kind of a yellow spot. It has no rods, so it's 100% cones. And the fovea centralis would be right in the very center of that. It has your highest concentration of cones, so really packed in there because this is where we're going to have our best vision. This is the slide of the retina. So this is figure 23.4. So we are going to have the retina here, the choroid, and the sclera. And the retina has layers. The first layer is the ganglionic layer. Here are the cell bodies of those ganglionic neurons, and you can see their axons coming together. And it's the axons of the ganglionic layer that all exit as a part of the optic nerve. This layer here is the bipolar neuron layer. So here's the cell bodies of the bipolar neurons. The dendrites are this way, the axons are this way. And this is kind of the middleman. And here we have the photoreceptive layer. So here we can see our rods. They're going to look like tassels, kind of very skinny rod shapes like pretzel rods. And then we have the cones, which are kind of triangular shaped if you use your imagination. So this is where we're going to convert light into a neural transmission, and it's going to open sodium channels and go through this way. So light passes through the ganglionic layer, through the bipolar layer, to the photoreceptive cells, where it's going to stimulate those photoreceptive cells and kickstart a neural transmission that goes through the photoreceptive layer, through the bipolar neuron layer to the ganglion neuron layer. The choroid and this little band of pigment here, their job is to absorb excess light. That way it doesn't bounce back and overstimulate these photoreceptive cells. Figure 23.5 is the dissection of a cow eye, so we can't do this one. Feel free to, if you have access to cow eyes, to dissect them. One of the nifty things about them, the tapetum lucidum, associated with the retina, also known as the tapetum fibrosum. This is a structure that humans do not have. This helps to reflect light so they can see better at night. This is what is responsible for eye shine. So when you use a flashlight to shine and you see a cat's eye glitter, or you catch the shine of a cow or a dog or something, that's what's reflecting that light back, is that tapetum fibrosum. Humans don't have that. That's why when we take a photograph, we get red eye. You're actually seeing the retina right through that pupil. Again, the pupil is just a hole. So a lot of different animals have eye shine. You can actually use a flashlight, go out in your yard and find spiders. Grass spiders have an eye shine. It'll be kind of a blue-green color. You could also Google the eye shine of spiders, or there's a video of somebody filming a spider covered in babies, and it looks like a little disco ball the way it glitters, but it's all the eye shine of those baby spiders. Figure 23.6, completely ignore, don't look at it. We're not going to do anything with that, or activity 4. When looking at exercise 24, activity 1, looking at the blind spot, you could do that if you want, but you're not responsible for any information about blind spot locating. The section on refraction, visual acuity, and astigmatism, figure 24.2 and 24.3, I'm not going to ask you anything in lab, but those are excellent pictures for lecture, because we do talk about the problems with refraction and so forth during lecture. Activity 2 is looking at visual acuity. Okay, so you would use the Snellen eye chart for that. So the Snellen eye chart, if I ask what is this, you would say this is the Snellen eye chart. And I can ask you what is it used for. This is for visual acuity or sharpness of vision. Your book doesn't have a picture of one because most people pretty much know what one looks like. That would be activity 3. Activity 4, that shows you the astigmatism eye chart. And that's this guy here, and that corresponds to figure 24.4. So if somebody has an astigmatism, this looks really weird. So normal vision, or people who do not have astigmatism, all the lines look the same length and the same thickness. But if you have an astigmatism, especially if you're not wearing your corrective lenses, these lines, some are thin, some are thick, some are short, some are long. And if you stare at it too long, they start moving. It's really trippy if you have astigmatism. Even though it may not be something that's very noticeable when you're reading, it does affect how you see things. People who have astigmatism, instead of seeing pinpoints of light when you look at a stoplight at night or you look at stars, we see little starbursts. So it looks very shiny and spiky instead of being a little pinpoint of light, which sometimes can make night driving a little crazy. Activity 5 is testing for color blindness. Again, you can look at the different types of eye color charts. They're usually called the Ishihara color plates. They're going to help you determine if you have red or green color blindness and so forth. Depth perception, binocular vision, figure 24.5 you can ignore, and activity 6 you can ignore. Activity 7 you can ignore. Activity 8 is using an ophthalmoscope. We do have them in the lab, but unless you've got one at home, you really can't do anything with it. So this is the ophthalmoscope. When you look at activity 8, that's the big word right there. Anytime you see OPHTH, that always means eye. Okay, so anytime we see that prefix, it means something to do with the eye. And this is just what we use to view the retina. Figure 24.6 shows you a picture using an ophthalmoscope. So I could ask, what is this? And you would say, this is an ophthalmoscope. I could ask, what do you use it for? You use it to view the retina. And that is it for the eye material.